I want to shift gears a little bit and talk about hit on another point that we spent quite a bit of time talking about a few years ago, the last time that we met during COVID, and that was the regulatory scene. I mean, do you recall some of our conversation around like what was going on regulatory in the regulatory environment during COVID?
Yeah, I think we did a good job predicting some things, if I recall.
I think we did. I don't know. We'll see. It was such an interesting time because, on one hand, I spent a lot of time talking to biotech execs at that time, during that time, who said, our clinical trials are challenged by, there was this challenge aspect. We're challenged by regulatory attention. Like we're challenged to get regulatory attention because there was so much attention being spent on the COVID vaccine and COVID therapeutics.
Mhm.
On the other hand, we were witnessing this rapid increase in the cadence of regulatory activity simply because we were in a, what is it? Would it be DEFCON? I think DEFCON 1 is more severe than DEFCON 5. We were in an emergency situation. So we talked a little bit about that when we last spoke. We talked about the speed and agility that we were seeing out of the regulatory authorities, and I believe you spoke in favorable terms about your interactions with regulators at the time. Do you feel like any of that speed and agility that was sort of injected onto the scene during COVID has stuck? Has it stuck around, or are you seeing more of a sort of return to normalcy? Like what's changed?
Yeah, I think more return to normalcy than than a continuation of what we saw. I mean, I got on the phone and for a particular call about a technology that was relevant to us in that period, and I remember there were representatives from BARDA and DoD and NIH and FDA, and I'm giving the presentation to these very senior people, like 18 of them from all these different interdisciplinary, departments and divisions across the government. Well, now our interactions are mostly limited to the project manager for our for our spinal cord program. So,
Mhm.
so definitely some things have gone back to normal. I think what we saw is what is possible, and I felt so proud in an extremely bipartisan way that what I saw was what America is capable of doing, whether it's the corporate side, the government side, the human side. It's why when I watch the Olympics and I see someone running and they're in sixth place, but they've got a USA jersey, I think they might just win this thing because there's something about the DNA in the United States that we find ways of solving problems when really pressed. But being human beings, we're fallible and we go back to the normal ways. We're not great at thinking about what statistical probabilities mean, and we fall back into normal human patterns and psychology. So I think a lot of things have normalized, and we've lost some of those special programs that existed for a very discrete purpose. But I do think that when we were talking, we spent a little bit of time specifically on cell and gene therapy. And I remember making a call that we need to see an increase, that the field is moving very quickly, and we need the FDA to continue some of its aggressive and rapid activity into cell and gene therapy, because otherwise it's going to get too far ahead of the regulators. And they have. If you go on to the FDA website and you look for cell and gene therapy guidance since 2021, there's a very long list of stuff there.
Mm.
So I think the push and the adoption of new regulations and understanding and expectations and criteria for monitoring and manufacturing and delivering cell and gene therapy product candidates has been able to keep pace in a very reasonable way. But when you're a government organization and you deal with an annual budget and you've got elections and everything, that's a very hard thing to do. So I think it's mixed. As it would expect it to be. It's mixed results. You're never going to go 10x and maintain 10x forever. It was great and encouraging to see that it was possible, but I also have seen the shifts into the right areas of what the agency believes is important and where the future is headed. And that's a very promising sign for us. We need that kind of regulation because it keeps the expectations high for the sponsors like us.
Hmm. To what do you when you look at that, that long and growing list of guidances from the FDA since 21, beyond your call for that on my podcast, which certainly I'm sure influenced the FDA.
I
I'm
appreciate
certain of it.
that.
Beyond that, to what do you attribute the attention that the agency has placed and the work that they put into creating those guidances and keeping pace
Yeah.
with the complexity and the technologies that sponsors are dreaming up every
Yeah.
day?
Yeah, the two things that come to my mind are one is sort of just simply supply and demand. I mean, there's this huge supply of new programs. The barriers to entry for gene therapy perhaps are not as, extraordinary as they might be in some other areas. And so you've got a lot of new programs. There's thousands of diseases and conditions to choose from. And so I think it is partly a response to the demand from industry. If we're coming forward with cell and gene therapy programs and, the regulators, that's what they need to staff up for. The other is complexity. Small molecules are well understood. There's established rules. Antibodies the same, but the complexity of a living cell is so many million times beyond that which we're accustomed to with small molecules that it requires new rules. I mean, literally new rules. How are we going to measure and control? You say you're going to make this cell. What defines that being that cell? I sometimes would say to people that we could make something that's completely synthetic as long as it's safe and effective. It doesn't matter if it's a cell that has properties that don't normally exist in the human body. That's not the criteria for a new product. The criteria for a new product is around safety and efficacy. So I think those are the two drivers there, is that there's been an incredible shift into cell and gene therapy. And so the regulators and everybody, the payers, everybody in the channel, everybody in that ecosystem has got to be responsive to that. And then, the old rules don't apply. If you apply, the standards of small molecules to making cell therapy, it's all going to fall apart. I would just joke the dose. I get asked almost every question I get asked about dose. And I always make the joke that, I don't know. I mean, if you put 100,000 cells or 100 million cells into a person, how many of them survived? 100 million molecules of aspirin go into your body. Well, we know what happens to them. But cells, they can divide. They might not survive the transplant. How many of them are going to graft and be durable? So dose immediately is different the second you push the plunger on the needle. So the old rules of dose response might break down in the cell therapy. So you can take that kind of analogy or comparison as far as we need, but I think that's really what the agency is responding to, is this is where the future is headed, and they need to be part of that and ready for it.
Yeah. Yeah. Um, you alluded earlier to the advantageous position that you're in, knowing what you know about where lineage cell therapeutics can go and perhaps will go. And obviously you can't divulge all of it, but I do believe there have been a couple of publicly stated preclinical programs added to the docket since we last spoke. So what can you share about beyond the two very active clinical programs right now? What can you share about what's coming next for Lineage?
Well, the two that I think you probably have in mind since we last spoke are the photoreceptor program, so we can manufacture these populations of photoreceptors. And being that we operate in the eye, photoreceptors is one of the other major cell types of the retina. And so it would be natural that we would, move one step to the right from the RPE cell to the photoreceptor cell. The other program is called ANP1, just for auditory neuronal progenitors. So this is a program. So we're making the auditory neurons that pick up the sound from the hair cells to help people who have hearing loss. I really like that program for a couple of reasons. In particular, it shares a lot of similarity with the dry AMD program, right? It's a relatively small number of cells. They go to a very targeted location. There's not a lot of competition out there. But as we just saw some from announcements from, there's a big pharma just announced recently that some wonderful success in modifying disease. And essentially the headline was, deaf kids can hear again. And that's really extraordinary. It shows you that this massive problem with sensorineural hearing loss can be modified with agents like ones that we are working on. And so that's really exciting. And this is a program that right now is in functional animal testing, right? So we're looking at hearing loss in animal models. You and I talked a few years ago, and this program didn't even exist. So what's really remarkable here with what we are doing is that we have the ability to skip a lot of those traditional small molecule steps. We don't have to develop an assay that we hope on a validated target, screen 5 million small molecules, find hits, develop those hits to leads, do structure activity relationship, and all the synthetic organic chemistry that goes along with it to try to finally get a small molecule lead that starts going into animal testing. We went from sitting around the table and deciding to go into hearing loss to animal testing in less than 1 year and less than $1 million. You cannot get that kind of efficiency. And so we have these really interesting advantages with our approach that I think ultimately are going to help this company be very successful, because the auditory neuron is known, right? Unlike a small molecule where you have to figure out, how to get into the pocket and inhibit this SH2 domain interaction and everything. We just have to copy what already is there. So we know what we're trying to make. The auditory neuron is deficient or destroyed. We're going to replace it and inject it into the ear. If it remains durable, if it remains functional, it may be able to provide activity in the form of hearing. And so our ability to move very quickly is one of the advantages, I think, of our particular approach.